Okay, so I'm going to next shift to cases of unknown etiology, and then sort of give you an example of where we have been able to find a pathogen, and then ask some of the questions about association, right? So this is Uttar Pradesh, Deoria, which is eastern UP, and this is where we set up a lab. So this is the hospital. We set up a lab there, and this used to be the old nanopore, so it's called the MinION. So the samples used to come in, they go through routine testing. So I'll tell you, so this was part of a big surveillance study, which is a 5-year study across multiple districts. And I think Dr. Ravi is coming, and I'm hoping he will talk about this particular study in more detail. But basically, what they had was an algorithm of what would happen to a sample at the district level, at a tertiary care hospital, and so on. As I told you before, this particular acute encephalitis syndrome, you will hear about outbreaks of AES, particularly in Uttar Pradesh, Assam, and these regions, mostly affecting children. And so first, when they have AES, what you collect is the cerebrospinal fluid. So you need to have the resources to actually collect the cerebrospinal fluid. So it's a spinal tap. And then check for JE, which is Japanese encephalitis virus. This is the most common pathogen. However, what we found was that in Uttar Pradesh, where the climate conditions are a bit different than Assam, you don't see that much JE. Also, a lot of vaccination for Japanese encephalitis virus is going on there. Okay, it's a mandatory vaccination there. And so that's because of that, we don't expect to see that much JE. And after that, the samples actually go through some tests for known agents, which we think are the top agents causing this problem in this country. If they are negative by all of these tests, is when we will take them up for sequencing. So I'm going to show you very little data from this study, but basically what we had was some 254 samples across 3 states in Northeast India, and this is back in NIMHANS. And most of these, you'll see these have some kind of etiology. Most of these 137 are unknown, some missing data, but mostly a large fraction is unknown. And this is sort of the bioinformatics, how we think about it. We have a pre-processing step. So basically, once we get the sequencing reads, we base call them. Usually, we'll take multiple samples and be sequencing them together. So at that point, we'll need to be demultiplexed. That is, you need to separate out based on the barcodes and adapters that you have put, which reads came from which sample. You remove everything that belongs to the human genome. I use a particular software called PORCHOP, or there are some others as well, to remove adapter sequences and split the reads. Then remove reads. So sometimes I do this, sometimes I don't. So reads that map to ribosomal RNA for a bacteria, in the beginning, I like to remove them and see what's in the sample, because these are conserved among many bacteria. So most of the time, they actually give you hits that are not real. They belong to the family, but not that particular species. So I like to remove them when I'm doing the initial looking at what's in the sample. But lots of people keep them because they can also be good identifiers and present in abundance. So then you remove low complexity reads with a length filter. In nanopore, because we have long reads, I usually set a reasonably high length filter. Anything less than 150 base pairs, I throw away. You can increase this or decrease this based on but some kind of length filter is good. It helps take away a lot of junk. Then for identification, I usually use Kraken or centrifuge, and I will tell you more about these 2 databases. Centrifuge for longer reads, they say, is a bit better, but both of them are k-mer-based techniques. They divide your reads into small k-mers and then look for them in a sort of database of other k-mers. So, and then I try to do a confirmation with BLAST. Sometimes this doesn't happen. At this point, I'll also try and extract out the entire genome if possible, confirm with PCR or some other test. So as soon as we get some hits from here, there are many different ways. The question that you're asking is the hit specific? Or is it could it have come from some other organism? How much of the genome is covered? Can I have some estimates of how good my particular hit is? And then get an amplicon, get the entire genome out. Sometimes you get this, sometimes you don't. Once you get it, then do a phylogenetic analysis to see, does it fit with where you're finding this virus or this particular bacteria or so on. So what we did was, with the 137 samples that I showed you, we ran Kraken and so on. And one of the things that we got that we hadn't expected was this primate erythroparvovirus 1. We got it in two samples. We got it both by Kraken as well as by centrifuge. And so then we got the entire genome of this organism out. It's a parvovirus not associated with, not seen in encephalitis in India before this, in meningoencephalitis, but it has been suspected to be an organism that causes meningoencephalitis in other countries. And, but no one has sequenced it from cerebrospinal fluid till now. So then we sequenced this. All of these cases are from West Bengal, but none of them were linked to each other. So these were not clusters, they were sporadic cases. We got the entire genome from all of them, and they sit in a particular, like, separate branch of the phylogenetic tree and into genotype 3b, and they sit with other sequences from Germany and Brazil and Ghana and so on, but they sit in a clade of their own. And if you look at smaller bits of this, not full genomes, then they sit close to genomes from fragments from Afghanistan. So what we found was B19V genomes. First, we detected by metagenomic sequencing. Then we did a PCR of 403 samples, detected in 13 of those samples from meningoencephalitis in West Bengal. Many of the positive samples were from children. This was identified with other pathogens. So this already breaks one of the criteria that we said, right? So it was present, and those 5 of those 13 had JEV, strep pneumo, and in some samples, 8 of the 13 had no known etiologies, right? This is the first report of B19V from meningoencephalitis in India, and these are the first genomes from cerebrospinal fluid. It's a small virus, about 5 kb, so it was easy to get the entire genome out. Most of the positive cases actually had good immediate outcomes. So 10 out of the 13 recovered and were discharged. We don't know if they have sequelae, what happened to them later. 2 left against medical advice and 1 person died, but the person who died was also positive for JE. Now, what is the implication of this? It was detected, so in samples with unknown etiology as well. It is a probable pathogen associated with meningoencephalitis in India. It should be included in the diagnostic panel for meningoencephalitis, especially in children. We need to assess the causal relationship, and it is unclear and needs to be evaluated. Most of the cases had good immediate outcomes. However, the long-term neurological consequences, they need to be looked at. And this is the team that did the work. Darshan also did part of the work. He's in the audience. I don't know if he's mentioned here, but part of the NIMHANS team. Okay, so moving on, what is the third case in which we are looking for doing pathogen discovery? This is not a novel virus itself, but it's a virus that seems to be evolving new traits. So these are large animal die-offs in South America. So these birds, they are dying. There are others. Farah can tell you more about this in more detail. But what has happened is H5N1, this particular virus has now had mutations, and this kind of animal die-off is also suggestive of the pathogen being able to transmit among this host. I don't know if they found confirmation of that, but it is suggestive of that. And we did a little bit of a deep dive into the data to look at how it's looking. These are the different kinds of animals that this virus has been seen in the current outbreak, that is from 2015 onwards. If you look at different animals versus different places, and the size of the circles is the number of animals infected. Now you're seeing a sort of outbreak, roughly, it's been I think, you can see more of the orange and the brown, right? So 2023, 2024 onwards, you're seeing lots of different animals, lots of different places, lots from the US, lot of report from the US, but we don't know how many different countries and animals are affected, but it looks like a large number. And so this is a good case to go sequence and see what are the mutations, how is it changing, etc. Human cases, very few. These are, I think, about 26 human cases, and not necessarily from this H5N1. There are others, related viruses also circulating, particularly in Southeast Asia. So none of those cases from around this region has been confirmed to be the same one, but the ones in North America, particularly, there's a huge outbreak in cattle that has people associated with it. So this is again, if we did not know that this was avian influenza, this is a very good case to go and look at what virus is out there. okay, likely to give you something. So this is a good case for pathogen detection. And the last one is viruses in other animals, and this sort of surveillance-based approach to look at what's out there. There's a lot of pushback against this when researchers do this. But basically, these are the different viral families and what has been seen in bats. There's also, I'm sure you've spoken about this already, but there's a database of bat-associated viruses. Very few of them have zoonotic potential, but the ones that do are of public health importance. So it is good to know what's out there. And I already spoke a little bit about what you do when something is out there. You are more prepared because you know a little bit more about things. What is our hope in doing this kind of viral pathogen discovery or in different kinds of animals is that you will have this repository that you will quickly be able to use to design interventions and screenings and diagnostics and so on. Right. Okay. So that's this part of the viral discovery talk. Any discussions around this that you want to have? Actually, there's a lot to discuss around this viral surveillance stuff, but I'm hoping that you will have that over the course of this meeting with people who are better qualified to deal with this than me. But I'm happy to start that discussion.
Chitra, I have one question. So, say you're sequencing lots of these different animals from different kind of samples and everything. So in kind of NGS space, everything brings down to the number of reads and everything, right? You get millions of reads, something which is very good and some are very few. But say you have very few reads for a virus which is of very high importance. How do you come around to that problem of, would you think it's in a significant or?
Yeah, so for example, this erythroparvovirus, we got only 2 sequences. But we saw that other people had said in other places that this may be something associated. And there were PCR diagnostics already. So we screened a much larger sample set to identify more samples. That's one approach, to find other people or other animals who are infected. And the other one is where you do amplicon sequencing and get the entire genome out. And there, because you're specifically targeting a specific region, you have a higher chance of getting it out than metagenomic sequencing, which tends to be a bit insensitive. Like, for example, with amplicon sequencing, I think it took us, we were able to get everything beyond 100x coverage in a single, like all of the 13 we were able to sequence. Whereas if you wanted to do that by metagenomics, you'd probably have to sequence much, much more. And we may never get it. In fact, other studies that have looked at this NGS picking up something and PCR picking up something, I found very poor correlation between the two. So yeah, with PCR, you know what you're looking for and that helps you. So usually, whatever you detect by metagenomics, if you can get the entire genome out using some other technique, that's really good. It gives you much more confidence. And then if you can go screen a bigger population, that's also good. Yes.
Hi. All these sequences are from the biological sample from which it is collected is from the animals that are known infected, right?
Not these.
Or these are surveillances?
These can be anything. Yeah,
okay.
They can just be tissue samples.
Is there a database of for what viral family, which type of sample is best suitable? Let's say if I have only blood samples, and only few that I can test for. Like that, is there a database or something that I can look for?
So I think in this database, they also give you the source. It's one of the metadata that's collected together. So basically any sequence is a bit useless if you, and we'll come to that now, is a bit useless if you don't collect the metadata associated with it. And the metadata is source, time, location, date, species, these kind of things. So it should be there, and people who are making the databases will usually take care of that.
Since we are talking about virus discovery and you showed multiple sources like from ocean water and animals, there was also this thing that I read, so I just wanted to hear your comments on it, about scientists checking for new pathogens in permafrost in Siberia. So I was wondering, whether you all were also looking into that, and if you could share something.
It sort of comes under the same rough bracket of looking at what's out there and what may come into human population. So some ancient virus discovery has also happened. Like people look at mummies, people also look at permafrost. Yes, but what did you want to?
Just on the potential of these ancient viruses and the scare that they might, cause mass extinctions.
I mean, that I don't know, but You have to do the threat assessment for something like that, right? So you have to look at what evidence do you have, what kind of virus is it. From what I understand, they found papillomaviruses. One of the things, recently they found some human papillomaviruses. They have through electron microscopy. Whether they can find the nucleic acid for that, I don't know. But these proteins, the coat proteins, were well enough preserved that they could look at the images and say, oh, this looks like the human papillomavirus. But that's as far as we have got. Very rarely can you reconstruct it, but ancient DNA work also happens. Why would you think about mass extinction with it? Like immunity in population? We'll have to think about it. We'll have to see whether.
But I think that, the thing about the way science works, it's always at the edge of exploration, right? We are into some unknown. And one can think of that unknown as a positive thing or a negative thing. Like you can ask a question, maybe nobody should study nuclear physics, because then they'll just go out and make a freaking bomb, right? But on the other hand, knowledge is everybody's right, and exploring the unknown is also a right. It's a human ethic or curiosity, endeavor. So that is why we have regulations, and all of us in this field, we crib about it, but we know that regulations, statutory clearances, these are critical checks, because in the past, there have been huge ethical violations, introduction of pathogens by anthropologists, all kinds of things which have happened, which are incredibly wrong. So we must adhere to those. We are not out of society. We must adhere to those ethical regulations. And as exploration changes, newer ethical considerations will have to come in. So I think that's how it is. I don't know whether I'm giving too much gyan, but.
I mean, you have the most experience of having to answer this question, right? Because, so the people who are doing this kind of work, they are asked these kind of questions, like what is the use? What will we do if we know? Is there this curiosity-driven thing? Where is it going to lead us? All of these. So
and also because you guys are all in this field, people will ask you, your cousin or, somebody, your relative or your friend may ask you, why do this? Isn't it too dangerous? Isn't this? So you should also think about it for yourself and think about what you feel about it.
I have one example where Dr. Yoshi, he's mainly working on influenza viruses. So we are aware of that Spanish flu was responsible for many deaths. And he proposed to US government that he want to reconstruct that virus again from the dead bodies. And he got that permission. And he reconstructed that virus to know the science behind that. So what is the purpose? That is important actually. What we want to achieve by doing those experiments, and how we communicate those things are.
So how many of you are familiar with the West Nile virus and the outbreak of West Nile virus in the US? Anybody? Okay. All right. So I want to, because we're going to start with pathogen genomics now, I wanted to tell you a story a little bit using the data to say basically how this kind of data can inform us. So this is a story, this is from an organization, a toolkit called NextTrain. Those of you who are not familiar with it, this is a very nice toolkit to get introduced to and work with. So basically, so you can ask the question, so basically there's an outbreak of West Nile virus. This is the first around 1999 is when you had an outbreak around the Bronx Zoo. I mean, later we know that this is around the Bronx Zoo. There's a book that has been written about it. There are some special features of this outbreak. So from there, from New York, it spread all over the US, killed all the crows, basically died, I think. So it impacted birds quite badly. And then it went to South America, and then now from South America, it's going back to North America. So this is the rough story and how we are able to put together this story using genomic data. So anytime that you're seeing this kind of link, it's based on the proximity of two sequences with each other and knowing the time as well as the genetic information about that sequence. Okay. All right. So we know that there was a big outbreak in New York, and then slowly it went all through the US, then into the West Coast, and then all of North America, entire region. Basically, based on the genetic evidence, you can look at the dispersal velocity about 1,000 kilometers per year. And in 2002, it was in the West Coast and was detected by the local surveillance systems there. What are the factors? It is likely the large population of naive and susceptible bird hosts. So when they looked, actually there were these vets in the zoo, the Bronx Zoo. They found that birds who had their origin in Africa or were brought from that region actually did not fall sick, whereas other birds were very susceptible. So in North America, there were a lot of susceptible birds. That was the first inkling that this was a sort of transported virus. And the virus then spread basically mostly by infected migratory birds, but probably there were other mechanisms as well. If you look at the phylogenetic tree, I think this one here is the outbreak in New York. And then after that, this was the only one sequence. I think this was from Israel or something. So this is the only sequence from the Middle East that is the closest. So it suggests that there was some link there. And then it spread all across the US, and there are split into multiple lineages. And there was also like sort of this establishment of local lineages in different places. So you look at, once you have the sequences, you can also estimate the substitution rate. So that's the number of nucleotide substitutions per site per year. Per site is per site of the genome of the virus. This is also a flavivirus, so roughly must be around dengue size. Right. So then, so basically over time, what are the mutations? This is the rate of mutations that accumulated, and you can see that at least there are multiple lineages coming up, and it has become quite different from the first sequences that were there. And then you can see that the virus also become entrenched, And so there is a persistence of some branches well over time. So there's this branch, it divides into many branches, but it sort of is there. And once you know the location, you can ask the question, was it present in that location? So all of these are New York. And so there are multiple lineages, all of them are present in New York. And then our cases evenly distributed throughout the US. So you can also ask about like how, based on the diversity, you can start asking how much of the population was infected and so on, and are there human cases and things like that. Then after this, there has been a spread to other countries, and then it sort of is also coming back to the US from South America. So this is the kind of analysis that we can do once we have the genetic information. So I wanted to use that. Okay, yeah. So this is the website, and you can also put your own data in here and use the suite that they have to do this kind of work. This is for Ebola. They did this kind of transmission, looking at transmissions, looking at lineages, looking at substitutions. A lot of this work was done for SARS-CoV-2 as well. So these are some of the resources that I wanted to tell you about that sort of fall under this bracket. So GISAID has a lot of SARS-CoV-2 sequences, a lot of influenza sequences as well. Outbreak.info also does a lot of analytics around this. If you want to look at some bacterial and viral bioinformatics resource, this one is a good resource. For Kraken, if you're using Kraken, there are already pre-made indices that you can use here. And that's really useful when you're doing classification to already have the indices. Otherwise, you need a lot of computational power to generate those indices. Then, yeah, this is a very good resource for going through all the steps of pathogen, like basically metagenomic analysis. They do a very good job of going through each step. And there are some other. Artemis is a good tool if you're working on bacteria and comparing across bacterial genomes. And there are lots of online resources of how to use Artemis as well, so I'll let you do that. So I don't know if this is of use, but we can just rush through this if you find it useful. Yes.
Different. Chitra, would it be possible some of the slides you think it's okay to be shared with the participants?
Yeah, all of them can be shared.
Yeah, and some of the papers or resources you recommend, we can share with all the participants.
Yeah,
something
sure.
like that.
Yeah, I mean, the slides, no problem. I just got a bit panicky about online streaming. Okay. Okay, so this we'll go through this. I just wanted to understand generally how many people are interested in like doing data analysis on your own. I need show of hands. Okay, then we should go through this. All right. All right. So in terms of terminology, these are the terms because this is meant like really basic bioinformatics 101 type. Okay, so what is a DNA sequence? What is it? This is what we'll be going through in terms of terminology. What is a DNA sequence? What is a nucleotide sequence? What is a reference sequence? What is a sequence? What is a read? What is a sequence library? What is alignment? What is assembly? What is coverage and depth? What are annotations? These are the things that you should be able to answer after we have gone through this. In terms of a standard workflow for DNA sequencing, what you will have is pre-processing, then alignment, usually alignment to a reference sequence. So this is not necessarily pathogen discovery type work. This is any kind of bioinformatics for pathogens, okay? Once you get the sequences, they can be amplicon sequences, they can be metagenomics. Irrespective, roughly there will be an alignment step. Then there's post-alignment processing, there's variant calling, there is annotation and interpretation. Instead of alignment, you will have classification for metagenomics into k-mers, like k-mer-based classification. In the pre-processing step, what we are trying to do is to clean and trim raw sequencing reads to remove low-quality bases and adapt a sequencer. This is what comes under QC. Alignment, these mapped and processed reads will be aligned to a reference genome. When you don't know the reference genome, you'll be using two different strategies. One is this k-mer-based classification, and the other is something called de novo assembly. And I'll talk about that a little bit. Post-alignment processing, you remove duplicate reads, realign around indels to refine data accuracy. This is post-alignment. It's called deduplication. There will be softwares like Trimomatic and so on. I'll give you a list of softwares that can do this. Variant calling is to see, identify genetic variants by comparing aligned reads to the reference genome. And there are some cutoffs that you'll have for this. How can I be certain that I've called a variant correctly? For that, there are some things that you need to know about that. Annotation and interpretation is basically how do you annotate variants with biological information that you have and interpret their significance in the context of your study. Usually, like, where did this mutation happen? What does that gene do? How does this mutation likely to impact that particular change, and so on. These are the file types that you will be working with. The first one is FASTQ. This format contains raw sequencing data along with their corresponding quality scores. Then there's something called BAM or SAM. These are alignment formats. So the first one is a binary format, the second is human readable. These are binary and text formats specially used to store alignment information, including mapped reads and their position in the reference genome. Is this going above, is this very high? Do you want me to explain a little bit more? You just need to familiarize yourself with these terms right now, okay? Because we can go over them a little bit more if you need to. Then there's this variant calling format. This basically has nothing but list of this position in the reference, this position is a G, in your sequence, this position is an A. That is what a variant calling file contains, okay? It has a position information, a reference allele information, and your sequence information. This BED, GFF, and GTF files, these formats are for storing genomic annotations, that is gene locations, exons, other features, and so on are stored in. So your primer binding regions, usually when you have a primer, you have a set of primers, you'll also get a BED file along with it. That tells you my primer binds from position 1 to position 15 of that particular genome. So you need to know which genome, you need to know position, and you need to know length of your primer. Those information will be contained in these kind of formats. FASTA, this is the most commonly used format for storing reference genomes, and this is nothing but a FASTQ file stripped of the quality information, okay? And each of these file formats looks a certain way, has a particular kind of headers, so that any software that is taking them will know how to use them, will know what to expect. So these are standardized file formats, okay? Summary of technology is basically Illumina that I've already told you about. These have raw data in BCL format, which can be converted to FASTQ using a tool called BCL2 FASTQ conversion tool. Then there are some Nanopore has got a FAST5 format, and it contains all the information needed for, like, that's the raw file format for it, okay? And then it has signal information generated during sequencing, and this FAST5 can be converted to FASTQ using base callers. So, and PacBio also has a separate format for itself. So the initial raw file format usually will be convertible to FASTQ so that you can do all the rest of your work using standard open source tools. In the case of FASTQ, how does each sequence look like? So each sequence in a FASTQ file is composed of 4 lines. The first line is the label. It always starts with an @ symbol, and it has a lot of descriptors. The descriptors may be anything. You can put anything. Usually these are machine generated. They are generated by the sequencing platform and they are put here. They are unique. Okay, so every read, so your read ID is the first line, always starting with an @ symbol. The second line is always a sequence. So this is your sequence information, okay? The third line is plus. Very rarely it will be plus some other stuff. It doesn't matter what it is. Third line will always have a plus, okay? And the fourth line is the base quality. Each base will have a corresponding quality score. Okay, all these these are called Q scores. They are ASCII characters. And how do you derive them? So basically, these are also called Phred scores. And you're looking it's called Phred 33, or when they give you your data, they'll tell you what to use to derive your quality, what was used to derive your quality score. And there is a list, there's a table that you can look up. What does it actually mean? A Phred score quality of 10 means the probability of incorrect base is 1 in 10. Base call accuracy is 90%. Similarly, if you're like usually Q20, above Q20 or Q30, you're looking at really low errors. Okay, nanopore data will be between 6 and 8. Just to give you an idea. But Illumina, we expect 20, 30. And nanopore is moving towards a Q20. So this is what people mean when they're giving you Q scores and telling you sort of averaging out. It's the probability of an error being in that sequence, okay? And this is how you read it. And each of these corresponds to a quality value. These are the QC tools. I will give you this so that you have it with you. So there's FastQC, there's Trimgalore, all of these. Many of these are like the popular tools. I've not put in everything that exists. And for nanopore, this PoreChop does a really good job, but you also have some other tools. We'll be looking at FastQC and what kind of statistics it's looking at, because there were some questions. Yes?
Sorry, naive question. So you said nanopore's accuracy is somewhere around 0.99, 99%?
It's worse than this. So, 1 in 5, can you?
Okay, I mean...
at some point, it becomes equal to chance, right? So that's bad. Anything over that, we say, okay, and we live with it. Nanopore per sequence read will be bad. But once you accumulate these sequences and align to each other, you'll get much better quality scores.
Right.
So you will get reliable data out of a nanopore. You just need to know how to use it and be careful about the interpretation. If you're looking for SNPs, that is single nucleotide polymorphisms, and you're using a single read for it, you've got to change from the reference. But you got a single read from nanopore. It's very unreliable.
Okay, so like if you average them out, it still is unreliable.
If you average them out, it's good. If you have 10 sequences and all of them have that same change, then you can rely on it, right? 10 I'm giving you as a rough estimate usually. But if you're relying on a single read, then nanopore could be wrong.
Right.
It's just something to bear in mind about how these errors influence your interpretation. Or at least this is my understanding right now. Okay, so when you get your FastQC, what you do is you take this tool, you put your when you get your FastQ data, when someone will send the data to you, it will be multiple lines of this FastQ data. So you'll have multiple lines of this. So sequence 1, sequence 2, sequence 3, usually millions of sequences will be there. Okay, so you're not, if you physically open that file in any notepad or any kind of text editor, this is what you will find, that there's a repeat of these 4 lines again and again. Okay, so, and a FASTA file is nothing but a FastQ file where the first line will always start with a greater than, it will contain some ID and identifier. The second line will be the sequence, the third and fourth line will not be there. Okay. So it's also a very specific file format, and a multiple line FASTA looks like greater than symbol in every alternate line, because that is the read identifier and the second is the sequence. I'll come back to that. Anyway, here what you will get is for when you're processing, so basically what you're then processing is millions of such reads, right? So you need to use software to do that. You can't look at each one of them by yourself. So when you're doing that, you can ask, basically what this is telling you is, gives you a percentage GC score, gives you sequence length, gives you total number of sequences, total number of bases, what was the file type, what is the encoding, because the encoding depends on how the Q was calculated, right? So here, all of the sequences are good because all of the Q values are above 20. Many of them are in this sort of 36, 34 range. So this we consider as good data. Now bad data looks like this. So this is position. So each of the reads is 40 base pair long. All of the positions are having good quality. That is how you interpret this. Here, 1 to 40 base pairs, half of the locations have very poor quality. Okay, and now when you're looking at this, what do you think you need to do? Yeah, so but what will happen is you will lose 20 base pairs and you will have only 20 base pairs remaining. This is something to bear in mind, that you can trim and you should trim because this is the reliable information, this is the unreliable information. However, you also lose data and it will impact your, when you do alignments and so on, you'll have only 20 base pair alignments. So your confidence in the alignments will be a bit lower and so on. So here, this is looking at again the quality scores, and you will see a hump in this when you're having some bases of bad quality. You can look at across the genome, what is the ATGC composition and things like that. If something is wrong, you will get to know. So these are the things to look at your entire data set level to see if something has gone wrong during sequencing. So this is how you would know that. Yes, Mike. Here.
Yeah, so that sequence, it's one stretch of base pairs, right? In the previous, this one. Yeah, it's one stretch.
So every sequence that is present in this data set is 40 base pairs long, or roughly 40 base pairs long. And on position 1, this is the Q score usually. This is the sort of average Q score. That's why you're getting this. It's average Q score of your millions of reads. Here, how many are there? There are, 250,000.
Yeah, but that 40 base pair together is one stretch.
40 base pairs, one read. Yes, yeah.
So why is it that first few are good quality?
So your polymerase is doing a good job in the beginning, then something bad is happening to it, some reagent issue, anything. This is very common, by the way, that the ends of reads are badly sequenced.
Okay, so it can change if you change the direction then?
Yeah, usually in the 5' to 3. Yeah, but both ends are usually bad.
Okay.
Do you want this?
So, can you take that?
Yeah, please.
So normally with Illumina, they first make clusters for your libraries. So by cluster, you mean that if you have one library, it won't generate enough signal or fluorescence from your DD NTPs to be captured by your camera. So you need to multiply it. So that process is cluster generation. So with every cycle, what you do is you put your DD NTPs and then you map, your camera takes a snapshot and then maps it back to X and Y coordinates from where it is capturing those signals. And this happens every cycle. How does it capture the fluorescence? There is a laser which bombards it at a particular wavelength. So with every cycle, you are bombarding with laser and then taking image. So when it happens over a period of time, from cycle 1 to 10, 20, 30, 40, 50, your fluorescence, your fluorophores start becoming weaker because you are bombarding them with a high intensity laser every time. So with progression of cycle, your fluorescence ability to distinguish itself very explicitly starts getting compromised. That's why at a later stage, we start getting bad quality data. Point 1, that's why in Illumina platform, you have concept of read 1, read 2. That's because when you do read 1, you capture say 150 times cycle, then your clusters start becoming weaker. What you need to do, you need to rejuvenate them. So you do reclustering again. using from the same cluster. That's why read 1 and read 2, because you need to rejuvenate them, provide some, redo the clusters again. So that's the technical limitation, or that's how it is technically placed. So if you have, for example, if they come up tomorrow with lasers which can be softer or fluorophores which are more stable, this problem will go away. And that's why there are newer technologies now in the market which are actually working on this domain. And one of them is like Element Biosciences AVT. AVT has come up with a fluorophore which is more stable than what Illumina is using. And they are more sensitive as well. So you need less intensity lasers where your fluorophores can be captured on them. So that's one of the limitations of Illumina at the moment, which is being harnessed by other technologies to come up with solutions as well.
I just have one more approach. I don't know if it is always 40 base pairs or...
this is a typical Illumina. In nanopore you won't, you will have very varied lengths.
Okay. Could you calibrate the number of base pairs in a particular gamer, or this, how do I call it? Like you do fragment and then you read them, right?
In the case of Illumina, yes. In the case of nanopore, no.
Okay. So could we reduce the number of base pairs?
If you reduce the number of base pairs, what you can do with it later will decrease. Long reads give you a lot of ability to align. Even if there's a poor alignment, it can have high confidence that it is this particular organism. So it comes at that trade-off. Okay. And really small sequences, then you have to stitch them together quite a lot. So,
but we do use adapters. Okay.
Okay, because this is coming from your sequence alone. You can get good data sometimes. When you get bad data, then you have to deal with it. And you have to know that it is bad data in order to deal with it. So there are other things to look at as well. And then if there is a particular k-mer that is enriched, like for example here, there is this CCCCC that is being enriched, it suggests something is wrong and that you should try and see in what context this is appearing, and you may see whether it's some adapter or something, or some region that you might want to remove. It may be part of here. So here, for example, there are this overrepresented sequences. Now, if you haven't enriched for something, then it's a good idea to see if these are adapters or something that you put in, because these are coming up too many times. It's just a QC, so you want to check why that is happening. So QC parameters and threshold may represent particular use cases. So for example, if you have an organism that is GC rich, then that percentage GC content may not be a flag that you want to pay attention to. So it's just something to bear in mind that might be specific to your case. So you must assess the quality as it's relevant for your project. Also, when you're doing detection versus when you're doing like assigning a mutation or something, the use case is different. So if you're just doing a detection, you may work with slightly poor quality data in the beginning in order to identify better, and then have other checks and balances in place to check things. And some QC thresholds may not be applicable to you. Now, in the case of alignment and reference, what happens is you have a set of reads, and you have a reference genome and you're mapping, and you will get read 1 mapping here, read 2 mapping here, and so on. And you will see that they're not perfect matches, that there are mismatches. Now, your file formats like SAM and BAM allow you to keep track of where the reads mapped, where were the matches, where were the mismatches, and so on. So this is what a SAM-BAM file looks like, and these are the different mandatory columns that have to be there and what each column contains, and so on. So these are, just to give you an idea that these are standard formats, and any time you get a SAM file, this is roughly what it's going to contain. This is the information. The first column is always going to be the query name. The second column is going to be a flag, and so on. So this is just something for you to know, that that's sort of what it is like. And in the case of what is a match, what is a mismatch, it has some particular rules, and you can look for these flags in order to identify them. And there's something called a cigar string. It tells you about the number of matches, number of mismatches, number of deletions, and so on. So knowing how to resolve this is also a good thing for you when you're looking for how good were my matches. There's this idea of depth and coverage. Coverage is nothing but breadth of coverage. So you have a genome, you're looking at the reads map all across your genome, then your coverage is what percentage of the nucleotides of the genome have got a read covering them. even one. So you can do coverage at 1x, 10x, whatever. So you can decide what is the x, and usually people do 1x for this, even if you have a single read mapping to a position. So why is the coverage 99.8 here? You can see that this particular region has got no sequences. So there is no coverage for this region. That's why you get a slightly lower number than 100. And what is depth? So if you take read 1, read 2, read 3, read 4, for each position, you're asking how many reads cover that particular base. That gives you the depth of sequencing. For example, there is an example here. For example, look at this position, TTA, right? Even though this is an A, the depth is 3 because there are 3 reads covering this position. The fact that they happen to be different will inform your variant calling and things like that. But the depth for each position is 3. So when you say, I want a genome at 10x or 100x or 1000x, this is what you're looking for. You're saying, on average, I want at least 1000 reads to cover each of my positions in the genome, and so on. Okay, so that's called depth of coverage. There's a way to calculate expected depth based on the total number of sequences and the length, where you make some assumptions about the uniformity across the genome, that you say that, okay, I expect that all parts of my genome are sequenced equally well, which is a wrong assumption, but if your primers are good, etc., you make that assumption when you calculate this expected depth. Then you say, how many reads do I need to roughly get a 20x coverage? And then when you're asking someone to sequence for you, you say, I need at least so many million reads so that I can get a sort of theoretical coverage of so much. Variant calling files, VCF files, they will have these column names. All of these are mandatory, okay? So there can be no information there, and usually this is how you read. If you get a VCF file, this is all the data that it should contain. It'll have chromosome name, chromosome position, reference ID, reference base, alternate base, quality of the variant, then other kinds of information that may or may not be there. What is a consensus? This is something that you can't work with the reads when you're doing phylogenetic analysis, right? You have to work with some kind of an aggregate of what is in your sample. So how do you do that? You do that using consensus calling algorithms. So these are ways to look at your mapped or de novo assembled genome and then say what sequence best represents all my reads. So there are many software tools that will help you go from this sort of mapped genome to a consensus. Where there is no information present, it will give you a choice of whether you should call the reference base or whether you should call N, and it's always prudent to call it an N. You don't know the information for that. This is something to be careful about, that you should look at what your consensus calling software does. Usually we use something called majority calling rule, in which we say anything that's present at greater than 50% of the reads, call it that base. So if it's A, just call it A. Leave the T alone. Where will the information about the T be captured? The information about the T will be captured in your variant calling file as a minor variant or something like that, if you have set your thresholds like that. Okay, so, okay, this is what a FASTA file looks like. Like I said, it has a greater than symbol, some ID, and then some sequence. This can be split up, or in reality, it is actually one line of like identifier, second line of sequence. This can be converted to this, okay? So when you're doing multiple sequence alignments, when you're comparing across different not different organisms, but different isolates and so on, what you get, it's best to use this format. Most of the softwares expect this format. And so this is an example of a multiple sequence alignment. All of these are different sequences or different consensus sequences. You align them together. Here they are colored as red, green, pink, green, blue, and what is the fourth color? Black, I think. I don't know. Anyway, so there are 4 colors here for the 4 bases. And so this is sequence 1, this is sequence 2. Then you get, basically what you're trying to do is you're trying to do an alignment of how many, like what are the least number of steps you need to go from one to the other. And there are many ways of doing this. There are many algorithms for doing this kind of alignment. The ones that are popular right now are Muscle. That's the one that I have used here. This software, it's called AliView. I think I list out all of these softwares later, but what it will give you at the end is a multiple sequence alignment. What you should do after multiple sequence alignment is do a manual inspection, because there may be some bad sequences with adapters or with something else that someone hasn't been careful about, and you should remove those before you do your phylogenetic analysis. I'll just touch a little bit on taxonomic classification, and then I'll stop and I think we can discuss. In the case of taxonomic classification, what you're doing is you have, what you get out of it is you take your reads and you bin them into known taxonomic categories, and then what you're able to generate at the end of it is a taxon abundance profile. Then you get a phylogenetic abundance, and you can get the coverage profile, genome sequence, what number of the fragments cover an entire region, and so on. So this is Kraken. In Kraken, what happens is you have a query sequence. It is broken down into k-mers. Each of those k-mers is then mapped to a pre-computed database. Those are called indices. Now these indices, it's better to take ones that are already done. To do the indexing yourself takes a lot of computational power. And I don't know if I have, okay. The link that I had given you is actually all the pre-computed indexes. So it's good to use those. But when the database changes, then the indexes also have to change. So just keep track of what you're using. For example, anything before 2019 will not contain SARS-CoV-2. So they'll all group to coronaviruses. So, so here, this k-mer maps here. All the blue k-mers are mapping here. All the red k-mers are mapping in these places, 2 different places. Now, when Kraken, this is particularly what Kraken does, it says, okay, so this is 1, 10, 4, whatever, then it's going to take this particular route. This is what it's going to say so you're going to say that the sequence will be classified as belonging to this leaf. And so it uses something called this, this route to leaf path is scored, and you get the best possible path or something like that. And there are many different ways, as you can imagine, of doing this. So it's mapped to the lowest common ancestor of the genomes that contains that k-mer in the database. So this is sort of what do you get at the end of it? You will get a list like this. I don't know if you can see this in detail, but this is basically saying unclassified. So there were a lot of sequences in whatever I put in that were unclassified. Of the ones that were classified, there were many that are in this sort of route. Out of them, some are classified as viruses. Within viruses, they belong to riboviruses, and so on and so forth, all the way up to Japanese encephalitis virus, but not just JE. There are lots of other viruses. So this looks like, if you look here, in the newer Kraken also tells you whether it's a unique k-mer or whether it is shared in that particular, all of the flaviviruses. So there is a way to look at that score here. And there is an abundance estimate that comes up in the first column here. You can also look at it as a chrono plot of viruses and so on, and then you can keep clicking and it will take you down this particular tree. This is a very common way of looking at metagenomic data, where you look at reads per million and you estimate abundances and make a heat map of that. So viruses are here, different samples are here, and then you can say, okay, how much, like what kind of enrichment did I get over the water sample or something like that, and calculate that apart from the reads. That is the number of reads that hit that organism in a million reads, normalized to a million. Yeah, I think that's it. And then these are the resources. I'll share it with you. This one is particularly good for all the steps that I've spoken about. A lot of what I have put in here has come from some of these. So I think that's it. Yeah, that's it.
I have a question. So say we are sequencing lots of things and everything is database driven, right? If it will map or come close to something, if it's in the database. Considering we are doing an exploratory work, there might be some reads which will be novel and may not map to the data, but they are very good ones. Some very significant mutations. How do we go about that?
Significant mutations?
Like, I mean the reads, I mean.
So basically, so now we're talking about, so basically whenever I've encountered these kind of reads, the problem has been they've turned out to be bad quality reads. So then you can junk them. You should have really, okay, these are my opinions. So I'll tell you, Usually when I get reads that are unclassified or don't map, right? Very few of your reads actually get classified. This is very common in metagenomic analysis. Now, if you've set really high quality filters, then you should look at what has not mapped and see if you can make larger contigs. That is, if you can stitch them together. This is where de novo assembly is used in viral discovery usually, because you're saying, okay, maybe this fragment doesn't match anything, but if I can join this fragment and make a contig, maybe it'll match something. And some viruses have been discovered using this approach. Also, if you now know what family you're looking for, then it's good to look for RDRP or some conserved protein signatures. So there are these particular, so the thing is, while Kraken and centrifuge have their limitations, the other thing to do is translated blast. So you take your sequences, you translate them into amino acids, and you do a match against protein sequences. That usually gives you a very good idea of the families, and some conserved things come up quite well. So that is used quite a lot. There are some other new tools also that are being developed with this idea that we should be able to place protein sequences a bit better. So can we work with those? And the idea is that your structural proteins, particularly of a virus, may have some constraints. So if you're a capsid protein, you have to look a certain way. So all capsid proteins will share some homology. So these kind of ideas, but all of them are pretty nascent and not very well used. I would say, and the thing that we are worried about in this kind of analysis, we worried very worried about contamination, whether it is there in all your samples or not. If it's in all your samples, likely to be some junk thing that you're not able to control, because we are doing so many ligation steps, forced ligation of molecules in this reaction, and we don't know what kind of chimeras they are producing. So if it has anything like your adapter sequence, you should throw it. So those kind of things. So the kind of quality thresholds you would have for something that's unclassified have to be very high. Once that is done, the one thing that Kraken and centrifuge are very good at is that even if there's a very small region that binds something known, that's a good place to start with. So there may be a 20 base pair region that binds a particular virus. That's a good place to start expanding from. And if you have the ability and enough of the sample, then that's where you build the primer and see what's near it and so on. That's another option. But it's hard. Like, I've been thinking about this a lot. Can never be certain that you're following the right path, and it really feels like it's a huge waste of time when you go after some small sequences. Like that Cardipiro virus, I thought about it for a really long time before saying, I don't have the sample, don't have a cluster of cases, don't know how to pursue that. It's 40 base pairs.
Other questions?
Ma'am, if you, now we have sequenced something, we have got a raw NGS data. Could you just let us know, briefly or vaguely, how to, come from the raw data to the actual,
From raw data, it has to go, all of what I've described here is exactly that. But the raw data, you should try and get it from whatever format the machine has into FASTQ. Once you have FASTQ, you go through all this pre-processing, assembly, whatever. It's just exactly what this presentation had, by the way. So I'm just surprised that you have this. Sorry, no. Maybe I'll understand it better once I show you this. Yeah. Okay. Does this answer the question?
So, so basically the thing about these kind of, it's nice to see a presentation, but until you do it yourself, it is not going to internalize, okay? And the good thing is, there may be no motivation, but a lot of these data are online. So really encourage you to try doing some of these things. I know sometimes computation can be a problem, and setting up the software, and then there's always some dependencies which are not there and all that stuff. But, let us know, we can always maybe point you to something or help out. But it's very good to play with data. If you don't play with data, it's very difficult to, when you get your data. Yeah. Any other questions?
Great talk, actually. This was really helpful. And so there's this thing, truncated data, right? wherein like what I realized was it because of bad downloading issue, but what else can there be?
Okay, this is a bit broad, but in nanopore, sometimes truncated data is something went wrong during capture, and then there are ways to retrieve that data because it's buffered. In the case of something that you're downloading, it just means that you didn't complete the download.
Yeah, so that might be the only major issue,
unlikely to be
right?
anything else that I can think of.
Okay.
Sometimes when data sets are very large, like for example, I'm trying to transfer some data of like 100 genomes. And it's such a large data set because it's mammal genomes that it's not possible to use FTP or something like that. So we have to, in fact, there's this thing called Globus, which is kind of a server developed by University of Chicago, and we have to kind of use, you might have seen vShare, what is that thing called? There are various other approaches to sharing data. Google Drive and all everything has its limitations. The minute we go into these zones where the data sets are very large, we're going to hit computational blocks. And I think nowadays, getting things sequenced has become relatively trivial. You just get the data from some company or whatever, you send them something, they'll send you something back, But then what does it mean? What do you do with it? That is a very, very different thing. And so I think it's really important to have that expertise to be able to go through these steps and check yourself every time. With anything you're doing for the first time, put in something which you know it should be this, right? And try it out. And make sure that you get what you think. Like even with phylogenies, you know that, how rats are related to mice are related to monkeys or whatever. So you try it out. And then you put in something unknown.
Doubt, ma'am, to clarify. So when we are doing the library preparation in Illumina, most time for homogenizing the samples population, we will be doing spiking with the 5x spiking. So what about, so when we are doing this spiking, most of the reads we were getting when the time of trimming, the spike X also we need to trim it out. So what is your idea when we are introducing a spacer, like a spacer primer to avoid the spiking? Is it will be better to use the spacers, introduce spacer primers?
Even those you'll have to remove bioinformatically, right? Do you have thoughts on this? If you have spacer primers instead of the 5x.
Someone in my lab has done it. I don't know too much about it, but I think Abhinav has done this, right? Put in spacer primers. Illumina. I think someone in my lab has done it, but that's for diet, metabolic coding for diet. I think it works partially, but you still have to you just retain more data. Because when you spike it with this 5x, you lose a lot of reads to that. So this is a better thing, but it's not like you can't get rid of 5x. But I can probably get a better answer to you after I talk to him.
Very basic question. What kind of extension does this FASTA file have, FASTA QC file?
It will say .fasta, but actually in reality, it is a text file.
It's a text file. Okay.
Many of these, see, whenever you get any of these files, it's just a good idea to just open them in a text editor and see what happens. Because many of them are like particular formats of text files. The BAM file is not. Yeah. That is not, that's a machine readable file, not a human readable.
A question coming back to basics, like I had this doubt. When we are, let's go to pathogen discovery, we have a novel data, nucleotide sequence, DNA sequence. What is, how do you classify it as something which is a new lineage? Or what is the significance, percentage of variation? And how does it vary from species or?
Yeah, do you want to take this?
Respond in the context of viruses?
Yeah. So basically, you just put it on the phylogenetic tree and have a look at it. And there's no actual real cutoff. The cutoff usually is if the phenotype changes. But there are some sort of rough rules about percentage homology and things like that. But they were, as far as I know, there is no cutoff that works across. Within a field, people may have an understanding of what I will call a new lineage. But in the case of SARS-CoV-2, there were some clear rules of there should be a cluster having that particular mutation, there should be a known epidemiological link, that is how they called it. I know it doesn't fully answer it because it's very specific to the case that you're talking about, like which pathogen, what is the history of the people working in that field, what have they decided, what is the nomenclature that they use, what do they call a lineage, what do they call a strain, etc. So it's very specific to that. You want to?
So basically, I think lineage is a relatively non-controversial term, right? Because lineage is in the phylogenetic tree. Does it cluster? And like if you have multiple samples, like, do they cluster together and not cluster with something else? Like, are they monophyletic? Are they in one lineage versus distributed across the tree or something like that? And lineage is not saying anything about species or strain. Those are more, I would say, qualitative terms. Lineage is defined quite precisely. Of course, the lineage could change when you add more data, when you add more samples. So that's the other thing, like we were discussing earlier, right? Like if you just download stuff from GenBank, you build a tree, depending on what you have, the whole thing could change. So that's why, again, I think, say you start with a paper, you look at what are the sequences there, you try to reconstruct their topology, whatever with download the data, reconstruct their topology, and then add in an unknown. Don't start with lots of unknowns, because then you will be completely messed up. You won't know where you're going. So start with something known, put in some unknown, one unknown, two unknowns, and then see how you can build up. But lineage is a correct term to use in, say, in a scientific paper or something like that, versus strain or species. Even species now, we can't really define it, right? I don't know whether someone wants to add to that. You want to add to that? Yeah,
It's a general thought. So, for like, you have any opinion on giant viruses, like Mimi virus? So it plays a very crucial role in evolution of very complex cells. So do you think it plays also a role in human evolution?
Okay, the only thing that I know about giant viruses is that you will see them a lot in metagenomic data sets because of the way the silica of the kit is made. It's because of that. Other than that, I haven't thought about them pretty much at all because they're just different from everything else. Other than that, no new thoughts to add.
Anyone else?
Is there no one else?
So, hello. So for this taxonomic classification, we often talk about OTUs and ASVs, right?
OTUs, yes, but ASV, I haven't.
Those are amplicon sequence variants. So basically, earlier what we used to do, like for bacterial classification, we used to consider like the sequence which are like so like close to each other. We used to club them and call it as one OTUs. Now, but there is new term, this ASVs. Now we are reading one sequence as one variant, like, and describing as like and we are classifying as we are considering one read as a one sequence, like, and we are classifying them. We are not clubbing them, right? So I often come across this paper which they talk about OTUs and ASVs. Some use OTUs and some use ASVs. So which way to go? Like, I wanted to ask.
Sorry, I actually don't know enough about this to have a thought about this, but you should, I think what I would look at in this kind of case is how many sequences cluster where. And if you have one or two sequences, then I would be a bit more concerned about the interpretation. If you have many of them clustering, then I have more confidence on that. So the abundance and enrichment would matter, right? But I don't have does somebody else know? Because I actually am not familiar with ASVs before this.
I think, as far as I understand, both are the same, actually. Yeah, ASVs and OTUs are actually used in the same context.
Just that the clustering mechanism is different.
Clustering.
Like how sequences are get clustered.
How they're defined?
Yeah, like before classification.
How OTUs or ASVs
Yeah.
Variant at all. Yeah, if there's a single nucleotide that's different, it's an ASV, but an OTU might be 1% different, 3% different.
So
right, so that's like you decided a cutoff of where to like cluster these OTUs, whereas ASV is just call everything that's somewhat unique in any way is an ASV. So it's just, yeah, your cutoff there for an ASV is one nucleotide different. I think so. Yeah, you could use them interchangeably, but yeah, just depends on how much data you want to kind of, or what your level of kind of lumping together you want to do.
I mean, I think that maybe, like, if that's the case, I hadn't heard of the term either, but given that we know so little about bacteria and viruses and stuff, at least when I see this microbiome data and all, I'm like, oh gosh, I mean, don't know anything about what those things are, but we do know something about, Acetobacter or whatever. We know roughly what that is. So maybe erring on the side of more like OTUs, which is more kind of, you roughly know which domain you're in, right? You know roughly where you are. Lactobacilli, something like that might be probably better than single base pair changes for taxa where we know so little. That's what I would say. Unless it's a very specific region like, like in viruses, binding site or something like that, where even one base pair is really important. Unless it's a functional region where you can attribute some change in protein or function or something to that particular mutation, and you have high confidence in it. This is assuming you have high confidence in that mutation, not error. Probably better to go with OTU just for taxonomic purposes. But that's just off the cuff response. I don't know whether anyone would like to qualify that.
Any questions?
So other question I had is, Chitra, about quality. So, RNA quality, because a lot of times when we go to these places like outbreak investigation or something like that, we're just going to get some sample and we can't really control the quality of that sample. So maybe your experiences or thoughts on that would be really nice.
Bad sample, bad data. That's it. I mean, it's just, we have tried to save it. Very rarely are you able to save something that is bad. But then we have better techniques now of stabilizing things, like, RNA later, some of these other things. But that's sort of my thinking. Like, we work around this a lot, but at the end of the day.
Yeah, I mean, based on experience, it's also a bit of what's the application we are looking for, because that's something which I always have my strong opinion on that say, for example, you want to do RNA-seq based investigation, and say you are looking for potentially RNA virus in that case. So when you make a RNA-seq library, ultimately your library size is around 280, of which your insert size is around 150. So ultimately we're looking at very small fragment of RNA. So if you try to outsource any work to a company, they will ask for mean value of 9. But that's not what you get in the field or clinical samples. We have now done few thousand samples work in the lab, and mean values of 4 or 5 also work quite well based on the application you are looking for. And this needs to be very clear when we are designing the work, that if you are taking a RNA-seq based approach, you are actually looking at RNA of 150 base pairs, and you are not sequencing single molecule of RNA. So you have, anyways, if you take 250 nanogram as a starting material, you have many templates of RNA available there, and you only need that size of RNA to make your libraries. So there are challenges, but there are solutions to it as well in terms of what application do we choose, point 1. Point 2 is whether you are looking for amplicon-based approach of whole genome sequencing, or you are looking for a shotgun-based approach of whole genome sequencing. If you're looking for an amplicon-based approach of sequencing, then probably your RNA quality becomes slightly more important than a shotgun-based sequencing approach, which is basically a de novo sequencing approach. So again, what application of sequencing you are taking for. Third, what platform you are going to use for. That becomes another determinant on it. So While asking these questions is also important to think of many other factors as well, to find a solution to the challenge as well. So yes, there are those challenges, but there is a possible solution in terms of what technique do we choose, what fragment length do we choose, what platform do we choose, and going downwards, what analytical pipeline do we implement. So if we take all these 4 or 5 combination into perspective, then possibly we may find a solution to the challenge as well.
I've just got a generic question for all of you, actually, the experts in the room. We are trying to figure out if we want to invest in short read sequencing or long read sequencing. So I'm currently debating between a PacBio machine and a NextSeq. So what are your takes on it? And our research is largely transcriptomics.
I'll let
To be very fair, between both of them, PacBio and I normally avoid naming the companies, but I normally don't go for PacBio for another reason is about data output from its SMRTs. So the data output is pretty much low when you are looking for sequencing decent number of RNA-seq. And normally when you go for RNA-seq based studies, then your numbers are will be at least around 50, 100, those, because then you are looking at case control and other parameters. Whereas in a PacBio SMRT, if I'm not wrong, data output is around 25 GB or so. Whereas in a NextSeq flow cell, with now X-sleep chemistry, P4 flow cell, you are roughly going to get around 500 GB data. So it's a balance between, what is my question? Again, I'm asking if you're looking for a transcriptome discovery for, say, a pathogen, which I'm suspecting something to be there in a limited number, then possibly I go with PacBio because it will give me longer reads with less number of amount of data. But if I'm looking for RNA-seq based approach for a case control study, then possibly NextSeq is a better choice in that comparison in that way. So it's again a bit of a subjective answer that what is the if I'm looking for de novo discovery of any potential virus or pathogen, probably going SMRT is a better way because you get longer reads but less data output. NextSeq, more data output but shorter fragments of 150 into 2. And I think now they have 600 cycle kit, but whether that's available with P4, which I'm not sure at the moment, but definitely they had with till P2 or P3, they have 600 cycle. So it depends on the question little bit, but if it is specific virus discovery or suspect you have, then use SMRT, it will give you longer reads. And if not, it's a case control disease study, then probably NextSeq is a better idea, especially with this new X-sleep chemistry where you are looking at 500 GB data. So you are looking at potentially doing 40 samples sequencing at one go. And human transcriptome I am talking here. So.